Yeah, too hot for anything. So I think that we'll probably go ahead and get started. Hello, everyone. Thank you guys for joining us today. My name is Amanda Moore, and I'm honored to co-host Dr. Garrett Nash from Memorial Sloan Kettering. And I really want to start by saying, how much I appreciate his time here today and all the work he's doing to help improve outcomes for cancer patients, including appendix cancer. So, like so many people here, I'm guessing I was just shocked when a rare cancer in general, but specific, even more so, a rare cancer came into my life. I was getting ready to turn 49, and my husband had just turned 50. So in January of 2024. My husband David was diagnosed with appendix cancer. At the time, we'd never heard of it. Like so many people here as well. I turned to Google for answers, and then from there, I relied on the kindness of strangers to help navigate what quickly became just a confusing world. It was like a different language. Um, So over time, I've felt the need to give back to this community that helped me. I've volunteered with organizations like the ACPMP, Can Collaborate, and I also founded a Facebook group called Appendicare, where I try to focus. Sometimes it goes sideways, but I try to focus and sharing new research, new treatments, clinical studies to support patients and caregivers impacted by this, really sucky cancer. So now, why we're here tonight. During registration for the event, you guys submitted great questions, very thoughtful, important questions. We've compiled them. Some of them will be answered during Dr. Nash's presentation and others right after he finishes. But we're also taking live questions via Zoom chat throughout the event, and we'll do our best to get through as many of these as time allows. But before we dive in, I want to introduce you to someone. I'm honored to introduce you to someone who's become a friend and who is just a powerful advocate of appendix cancer. Um, Chrissy Buttigieg. Sorry, Chrissy, I know I butchered it.
You did so good.
Thank you. So Chrissy is a 10-year appendix cancer survivor, a patient of Dr. Nash, and a co-founder of the Facebook group Appendix Cancer, among many other things, mom, wife, sister, daughter, all the things. So we both believe really deeply in the power of community, specifically this community, because it's helped both of us so much. And that's why we've kind of combined our groups to bring this event to life, and I'm hoping we'll do many more down the road. But now I will turn it over to Chrissy so she can share her journey.
Well, thank you so much, Amanda. I'm so happy to be here tonight. So I'm just going to take a couple seconds here to introduce myself. As Amanda said, my name is Chrissy Buttigieg. I am a 10-year appendix cancer survivor. I was diagnosed with stage 4 disease in 2015. At that time, I did 10 rounds of FOLFOX, and then I enrolled in the trial Dr. Nash will be speaking about tonight. That trial, called ICARuS, was comparing the effectiveness of HIPEC and EPIC for appendix and colon cancer patients. During surgery, I was randomized to receive HIPEC. Unfortunately, 9 months later, I was diagnosed with a recurrence. So at that time, I did another surgery and I had 3 rounds of EPIC. So I've actually had both of these treatments that were involved in the trial. Next month will be 9 years since that last surgery and treatment, and I'm thrilled to say that I have no evidence of disease. Remission has afforded me the opportunity to become a patient advocate, like we were just discussing. I have been on the Patient and Family Advisory Council for Quality at Memorial Sloan Kettering for the past 4 years. Dr. Nash actually serves as our physician champion, so he really can't get rid of me in any context. Additionally, I serve on the Cancer Policy and Advocacy team for Cancer Nation, and I served a term on the Patient Advisory Board for ACPMP. It is my privilege this evening to introduce you to Dr. Garrett Nash. He is trained as a colorectal surgeon and surgical oncologist and was recruited by MSK to start the appendiceal and GI peritoneal program. He quickly found an MSK medical oncology collaborator, Andrea Sirsic, and together they have built one of the largest such programs in the US. He is the primary investigator of ICARuS, a randomized clinical trial studying the efficacy of intraperitoneal therapy for colon and appendix cancers, and that is a true mouthful. Last year, Dr. Nash became the Vice President of Quality and Safety. His goal is to make MSK a leading example of how a cancer center can offer not just the highest quality care, but also the safest care. He and Dr. Sirsic continue to lead the peritoneal program, aided by their clinical and research partners, Yorgos Karagunis and Michael Foote, and their laboratory science partners, Naikang Chung, Darren Veach, and Karun Ganesh. I hope I got those all right. With that, I will turn it over to Dr. Nash.
Hi, well, thank you, Chrissy. It's been a pleasure to have both treated Chrissy and now collaborated with her with respect to advocacy over a number of years. And it's also been great to get to know Amanda. I'm just looking at the attendee list. I can't see your faces, but I see names, a lot of whom are quite familiar to me. I think at least 6 or 7 patients, other patients of mine are here. So, thank you all for joining, those who know me and those who don't know me. I'm now going to share my screen. Amanda, I don't have privilege to share my screen. So I'm sending you a request to share my screen. Okay, that might have worked. Let's see. Okay, here we go. Sorry, I have a lot of windows open. Okay, here we go. Okay. Okay. Chrissy, can you see okay?
I see it.
Okay, great.
I think
so I'm going to talk about the design.
You might want to make it bigger. You might want to make it bigger because I can see your next slide.
Oh, you know what? Sorry, I'm sharing the wrong one. Let me stop sharing and let me share the correct one. Sorry about that. Just one moment. Okay, how's that now?
Yes, that's good.
Okay, great. So I'm going to talk about the design and conduct of the ICARuS clinical trial. This photograph, for those of you who may not recognize it, is actually a beach, believe it or not, that looks at Manhattan. It's called Governor's Island. This is a number of years ago. It was while the new World Trade Center was being built, and it was over 100 degrees that day. So everyone's sitting in the shade and no one's out here in the sun. But that's what that background is. I have no disclosures with respect to conflicts of interest. So I'm going to spend a little time not only talking about the findings of this study, but also why a clinical trial is such a powerful way of determining whether a treatment is effective. So we talk about hierarchy of evidence, where the lowest level of evidence is at the bottom of the pyramid, and the highest is at the top. And essentially, what this reflects is how much bias can be included in the findings of the report. So expert opinion is literally that. It's someone's opinion, and it obviously can be based on many factors, many of which are not particularly scientific. So we find that this is the most susceptible to bias. Retrospective case series are when you look back at how patients did after you treated them over a number of years to report what the outcomes of that treatment was, but it's not got any control. You don't know what would have happened if they didn't have that treatment. Case control studies are similarly looking backwards and looking at outcomes of a group when you try to compare them to another group with a similar problem, but were treated differently to look to see if there's any difference. However, these groups may not be equally balanced. It may be that, for example, many case control studies, sometimes the treatment group turn out worse than the group who didn't get treatment, but that may be because the treatment group had the worst cancers, and therefore they're the ones you treated, whereas the ones where you maybe used less treatment had less aggressive cancers. So as a result, they had a more benign picture to start with. Therefore, it looks like that the treatment was actually ineffective, whereas it actually might have been effective. Then is the prospective cohort studies. This is where instead of looking backwards, you offer two different pathways of treatment going forwards, and then you compare the outcomes, but they still may not have balanced groups. There still may be a bias why someone got one treatment versus another treatment. The data may be more accurate because when you look backwards, you're more likely to have missing data or perhaps incorrect data. But when you go and follow them forward in time, you sort of can have cleaner data. But there's still the problem with bias. So RCTs, the top, which really represent the smallest proportion of all studies because they're difficult and expensive, and you'll see why. But randomized controlled trials are considered the pinnacle, the most scientific way of determining what the answer to your particular question is. There's also this bubble up here, meta-analysis, which is when you actually combine the results of several trials together. If you find several trials that are structured similarly, using the same treatments and the same type of patient, where you can kind of look at the results together and get more power in numbers. So that's why meta-analysis is up here. So just to get into really some fundamental concepts of what is a clinical trial. So the Declaration of Helsinki, which came out of a lot of the unethical things that happened in Nazi Germany, when a lot of Nazi doctors experimented on people without any form of consent from the individual. The Declaration of Helsinki came out with recommendations of really how to conduct clinical trials in an ethical way and in a way which are most likely to get the correct answer. So a clinical trial is a scientific experiment involving human subjects in which the treatment is initiated specifically for therapy evaluation. The goal is to detect a soft signal in a noisy environment. And what do I mean by a noisy environment and a soft signal? Well, if you look at the opposite, so if you look at a crash test, like where they, Ford will crash a car into an object to figure out how safe it is in the setting of a car collision. So if you look at the crash test dummies and the bodily injury that they experience in a crash test, it's much more likely to relate to the intervention, the airbags, than anything else, or whatever else you're testing, seatbelts, airbags, as opposed to, an oncology study where you're looking at cancer recurrence, where it's got much more to do with, there's much more to it than the intervention, chemotherapy. In fact, it's much more dependent, the outcome is much more dependent on the type of cancer, the extent of cancer, the aggressiveness of the cancer, what we call the biology of the cancer. The cancer treatment is actually much less related to the outcome than the cancer itself, which is quite different than a lot of these other scientific experiments. So the purpose of a randomized trial is to eliminate bias, and we do so by randomizing patients. And randomizing patients mean that if there's two treatments in your trial, like in ICARuS, it is the equivalent of a coin toss to decide who gets which treatment. So there's no other factors that are going to play into it, like what the patient wants, what the doctor wants, or anything else. It's just the only difference should be the treatment. Also very importantly is the treatments are standardized. So I'm sure many of you have noticed from your reading and from talking to friends that the way we treat cancer can vary very different from center to center. Sometimes it's the dose or how frequently we give it or how much of we give it. With chemotherapy, there's a lot of variability in how you can actually administer any particular chemotherapy. And also the other important part is standardizing the detection of the outcome. So, some oncologists will get scans every 3 months, every 4 months, every 6 months. And if you're looking for small differences in outcome, it could make a a big impact if you wait an extra 6 months to get a scan, because that can be the difference between having a recurrence and not having a recurrence. And sometimes clinical trials can only detect differences if they're bigger. And sometimes these smaller differences can be very difficult to detect. So you need to have a standardization of how you follow the patients, with what tests, and how you define a recurrence. These are all things that you can, when a well-constructed randomized trial should be able to address. So going back again to the crash test, so an ideal experiment is use identical subjects, which are cars and crash test dummies. Every crash test dummy should be the same as every other crash test dummy, unlike humans, where we're all very different. So when you're comparing, let's say, a Toyota to a Ford in a crash test, they don't need to crash a lot of cars to demonstrate that one is safer than the other. Whereas clinical trial people are far from identical. We're different in age, gender, medical conditions, overall physical health. There's so many differences, and the cancers themselves are very, very different. And many factors that affect outcome of the cancer we still don't know. Like, why do two people who go through the exact same treatment for what looks to be the exact same cancer, so one patient is cured and the patient isn't? Why is that? Why did someone recur early and someone recurred 5 years later? We just can't always understand that. And measurement error can be pretty significant. So, for any significant trial, you need at least hundreds of patients. And if you're looking for a smaller difference, you need thousands of patients. So this is a very expensive proposition. So, for example, ICARuS cost about $2 million to run from start to finish. And as you all know, with rare cancers, it's hard to get funding, and rare cancers are rare, so it's also can be hard to find patients. So that can also, that's another challenge with clinical trials. Many of you are aware there are different phases of clinical trials. So I just want to talk through what those different phases are and what the significance is. So a phase 1 trial is looking at toxicity, and it's answering the question, is the drug tolerated or too toxic? And at what level of treatment does it become too toxic? And when we look at success rates of phase 1 trials, only 4% actually proceed to phase 2. So 96% are too toxic, or there's some other factor that prevents them from proceeding to phase 2. So if you get to phase 2, now you're looking at efficacy. Now, there's two terms that are used sometimes interchangeably, which aren't truly interchangeable, and that's effectiveness and efficacy. So efficacy is trying to show a response to treatment. So the classical example is you go on a new drug that's already gone through a phase 1 study, and you're looking to see if you can shrink a tumor. Not if you can cure the patient, not if they live longer than other patients, just will it shrink the tumor, or will it lower some blood measurement of tumor activity? And fewer than a quarter of those are successful to the point where they proceed to phase 3. So now we're talking about only 1% of all trials that go to phase 1 will actually go to phase 3. Phase 3 is looking at effectiveness. And rather than just looking if you can shrink a tumor, we're actually looking at reducing the risk of recurrence and, most importantly, prolonging life. Now, the numbers are much better. About 60% will proceed to drug approval. But again, if you now look, that means only less than 1% of drugs that go into a phase 1 trial will actually proceed to drug approval and will become available to patients. There's also phase 4, which is the least talked about phase, and this is real world performance. A clinical trial is an artificial environment. It's very controlled. You have to have specific criteria for entering into the trial. And if you're to think about this purely scientifically, the results of the trial only apply to the people who are in the study. So if you weren't in the study, you weren't exactly like some in the study, the results may not apply to you. And that's what we call generalizability. So we do need to for two reasons. One, to see that it's truly effective, because that it's actually be able to be used in the general population with results that are comparable to the results of the clinical trial. And also, is there long-term toxicity is the other phase 4 part, because these phase 1, 2, and 3 studies typically are completed in under 5 years. In fact, most chemo trials, the goal is to be finished in less than 3 years in a phase 3 trial. As you'll hear, ICARuS took a lot longer than that, but that's sort of expected with rare cancers. So phase 4 is real world performance. So before you start a clinical trial, you have to start with the research question. And the question should be boiled down to these 4 elements: the population of patients who are going to be in the trial, the intervention, the therapy you're trying to understand if it's effective, the comparison, the other treatment, which can be a control in some cases, like a placebo or no treatment, and the outcome. And so the question for this is, do HIPEC or EPIC reduce the rate of recurrent cancer in patients with appendiceal or colorectal cancer, metastatic to the peritoneum, who undergo CRS? So you break down the elements. So this is the population: patients with appendiceal or colorectal cancer, metastatic to the peritoneum, who undergo cytoreduction. The intervention and the control are really, or the comparison are really interchangeable here. In this case, we can call the intervention HIPEC and the control is EPIC, but really they're interchangeable. And the way we designed the trial is a way to see if one reduced the rate to a greater degree than the other. And the reason why there wasn't a control arm, a true control where no one got treatment, was because at the time we felt that patients and oncologists didn't necessarily believe that it was acceptable to have no additional intraperitoneal chemotherapy after cytoreduction. And our concern was if we had a control arm, it'd be very difficult to accrue patients. And that was also based on real life experience. The first HIPEC trial in the US randomized patients to getting HIPEC and cytoreduction versus systemic chemo alone, and they were only able to accrue 1 patient and they closed. And this is a well-funded, NCI-funded trial, and they couldn't pull it off. So our feeling was that we could not have a placebo arm. And then the outcome is reducing the rate of recurrent cancer. So why is this trial necessary? Well, I think, most of you are aware there's a lot of preliminary data based on these retrospective studies, expert opinion, case control, various studies, even a couple of prospective non-randomized studies that could make an argument that these interventions are effective. But really, they're just sort of what we call preliminary data, and they're not something that is scientifically proven. And there are a handful of actual clinical trials that sort of hint at some of the answers we're looking for. So I'm not going to focus on the preliminary data because there's so much of that, but I'm going to focus on the previous trials. And these are largely in colon cancer. The first one, the Dutch randomized trial, was a trial published back in 2003, so over 20 years ago. And it was the first trial to really look at what we call regional therapy. So intravenous chemo or oral chemo are generally called systemic therapy because it goes through your whole body. Regional therapy is just when you do a region, and the region in this case is the abdomen, and the intervention was cytoreductive surgery with HIPEC, and then they also got IV chemotherapy compared to just IV chemo. The population was patients with colorectal or high-grade appendiceal cancer. They didn't give us the exact breakdown, but the large majority did have colorectal cancer. It could be either primary initial diagnosis or someone who recurred in the peritoneum, and they couldn't have any other sites of metastatic disease. It had to be peritoneal only. They started with no chemotherapy, and so the randomization occurred before any surgery, and one group got cytoreduction and HIPEC and up to 6 months of 5-FU leucovorin. This was an era where we didn't have FOLFOX and FOLFIRI and bevacizumab and various other systemic therapies. So this was our only known effective therapy for colorectal cancer. So this was what was considered standard. And so the control arm was just that standard chemo. And then if patients needed palliative surgery because they had an obstruction or they had some symptom, they could have palliative surgery, but they weren't eligible for cytoreductive surgery. And this concluded there was a benefit. Now, often people call it a benefit of HIPEC, and even in their survival curve, and I'll explain what this curve, I'll walk through this curve to help people understand, to interpret these, because we're going to show a number of these, and this will be an important concept. This is called a Kaplan-Meier survival curve. But here they labeled the experimental arm HIPEC and the other standard. In reality, this was actually HIPEC and EPIC and systemic chemotherapy versus just systemic chemotherapy. So if you're not familiar with these graphs, the survival curve, this is the proportion of patients who are alive. So at the beginning of the study, it's 100%, and it can go down obviously to 0%. And then this is the time, so 0 months up to 36 months. They show the number of patients here. So there was 51 in the HIPEC cytoreduction arm and 54 in the chemo-only arm. And you can see as time goes by, these numbers drop as patients have recurrence. And what you see here is a separation of the curves, which is what you're looking for. And there's a statistical method of determining if this is really a meaningful difference with respect to from a statistics, or it could just be chance. And this met that threshold. So if you look at, for example, 24 months, about 20% of the patients who had chemotherapy only were still cancer-free. Oops, sorry. Whereas at that point at 24 months, you can see about 40% of the patients who got cytoreduction HIPEC. So twice as many patients were cancer-free. So this was a very compelling argument that this therapy, this combination, was superior to chemotherapy alone, and really has led to this era that we're currently in. What also was quite worrisome about this study was among the 51 patients, 8 died from complications of treatment. 8% died. No, sorry, 8 patients. Yes, so 16% died from complications of treatment, whereas none in the standard arm died from complications of treatment. So it was quite worrisome that the therapy was dangerous. Now, these investigators have shared that they were very early in their experience of doing this surgery and this chemo, and therefore it likely, and since this era, their complications leading to death rate has dramatically dropped. So it's definitely become safer, and that's, of course, led to its widespread use. There's another less known trial conducted in Sweden where similar patients, appendix or colon, again, no chemotherapy. They were randomized from the beginning. And here they got cytoreduction and just EPIC using 5-FU was the drug, they administered EPIC, and no systemic chemo. This was in a period where FOLFOX was now available. So the control arm was FOLFOX. And in this study, you again see separation of the arms. This is the blue line is the cytoreduction and EPIC arm. And the red line is the FOLFOX arm. It's a small study. This is just about 40 patients. So there's still a possibility that this difference is by chance, because each of these X's or circles represents one patient. So you can see by the end, there's only a handful of patients in this arm. And the separation only occurs at around between 2 and 3 years. So it's unclear if this didn't really convince anyone. In fact, the study was kind of abandoned because the HIPEC study had come out while they were conducting this study, so they kind of abandoned pursuing EPIC any further. More recently, so this was published or reported in 2018, 7 years ago now. This was often referred to the French trial or Prodige 7. And this was in the era, again, of FOLFOX and more experience with cytoreduction. And these patients were randomized to either getting HIPEC or nothing additional. And they had a pretty good number of patients, 265. They had to have carcinomatosis from colorectal origins. No appendiceal patients were included here. And the surgery had to reduce, no visible disease could be left greater than 1 millimeter. And then they were randomized to either getting HIPEC or no HIPEC. And here you can see the green line did not get HIPEC, but they had cytoreduction and systemic chemo. And the red line did get, in addition to cytoreduction and systemic, they also got HIPEC. And these lines don't separate. They're identical. There is absolutely no difference. And when you apply a statistical test it essentially says there is a greater than 99% chance that this is the real answer. So this really put a major dent in the perception that HIPEC was effective, because this was a very experienced group that really, before this study, they published a lot that indicated they were really convinced HIPEC would be effective, and unfortunately, it was not. So we started ICARuS about 5 years before this trial came out, and it was for both colon and appendix. But based on the results of this, we stopped the colon part of the study. And in order to make sure that we had enough patients to really get an answer for appendix cancer, we extended it so we could get up to over 200 patients with appendix cancer to try to answer that question. I'm not going to share the colon in interest of time. I'm not going to share our colon cancer data, but it was also negative for difference. So here we are with ICARuS. So here, the patient's not mentioned here, but it was the judgment of the surgeon and the oncologist whether to do chemo before surgery. And then candidates for cytoreduction were taken, and if we could get down to less than 2 millimeters, they then could be randomized, and this occurred during surgery. We essentially opened the envelope, indicating which treatment they would get during surgery. And they got either HIPEC using mitomycin C or EPIC using FUDR. And we used the protocol, the HIPEC protocol we used matches the most common one in the United States. We took basically the protocol used at UPMC and copied that so that we wanted to use a therapy that was most consistent with what most people in the country were getting. And EPIC was using our standard EPIC, which we developed here, more than 2 decades ago, more like 3 decades ago. So this was, we had a lot of experience using this. We have something called stratification, not terribly important, but basically we wanted to make sure the 2 arms were balanced, so that when we look at the end, that would have equal numbers of appendix cancers in both arms, equal numbers of colon cancers, and equal numbers of patients who had chemotherapy in the previous 6 months, because that made sure that what we thought were the most important factors in their outcome were sort of balanced between the 2 groups. And we didn't want to rely on randomization alone to get that balance. So 107 patients were randomized to get HIPEC. We administered it for 100 minutes with temperatures ranging from 41 to 43 degrees. We gave 2 doses of mitomycin. At the beginning, we gave 30 milligrams, and then 60 minutes into it, we gave an additional 10. Excuse me, I just need a little water. Sorry. So that was how we administered. And again, this was based on the Pittsburgh protocol. And then patients who got EPIC got fluoxuridine, our standard 1,000 milligrams per meter squared. So it varies depending on the size of the patient. And they got it for 3 days in a row. And more than 80% started it on the first day after surgery. We did allow for later starts if patients were experiencing some issues that we were concerned that EPIC might increase the risk of complications, or they wouldn't tolerate it initially, because sometimes patients were quite distended and uncomfortable after surgery, and we felt that they wouldn't be good candidates to get it, so we could allow room to wait. Every single patient got EPIC. They received it while they were admitted to the hospital, but again, more than 80% got it on the first day. And these are what the 2 groups look like. So this is the HIPEC group. This is the EPIC group. So their ages were similar. Average age was 56 in the HIPEC group, 58 here. Balance with respect to gender, there was more women than men in this. 65% were women in the HIPEC, 61 in the EPIC arms. And the race was pretty much reflective of our typical population of patients at MSK. And they were also well balanced, as was the ethnicity data we had, which is Hispanic, people identified as Hispanic. And then we also, thankfully, had a nice balance of the grade or the differentiation. So WD is the well-differentiated, the low grades. Moderately differentiated is the intermediate grades. Poorly differentiated are the high grades. And you can see they're well balanced in the two arms. The percentage of each is quite similar. They're also balanced based on whether they had, we define pre-op chemo as any chemo they got in the previous 6 months. So a little more than half the patients had chemo in the previous 6 months. Some patients had chemo before that, and some patients, particularly the well-differentiated appendiceal cancers, we did not routinely use systemic chemotherapy. So that's why not everyone had chemo in the previous 6 months. The PCI, which is the amount of cancer, I think a term most of you will be familiar with, but that's sort of the quantity of cancer we find at the beginning of surgery, was also quite similar, with the median, which is kind of like an average of 14. And as you know, the numbers go from 1 to 39. Generally, if it was over 20, unless it was low grade, we felt that was probably not appropriate. So that's why the numbers were around 14. And we achieved CC0 in 100% of patients. So these were all reduced to no residual visible signs of cancer. So we randomized 212 patients, 107 to HIPEC, 105 to EPIC. Then they all went into follow-up. Time we last looked, 60 patients had completed follow-up and had no evidence of disease out of the 212. 21 had no evidence of disease, but were still in follow-up. They're still in that 3-year window. And 129 had gone off study, mostly for progression of the cancer. A couple of patients moved out of the country and it became too difficult to follow them. And 2 patients went off, 129 went off study for progression, 2 went off study for other reasons, which in both cases was they moved to internationally and just was no longer practical. We did consent 54 patients for the trial who we decided not to randomize. 52 of them were found to be ineligible either with further imaging. For example, they were found to have liver mets or lung mets, or had some additional testing indicating that they had some heart disease or some other reason why it wasn't safe to go to surgery. And only 2 patients who actually consented to surgery withdrew and decided they changed their mind and they didn't want to be in the study. And here are the results. So what you can see here is that there's no difference between the arms. We look at the 3-year mark and it's 32% in EPIC and 30% in HIPEC. And I realize this may be quite stunning for a lot of patients to hear that HIPEC may not be effective, but that's what these data point to. And I understand many of you have had HIPEC and may believe that that's what saved you. But it may be very well other factors, including the surgery itself, chemotherapy you may have had, or something about your cancer that made it less aggressive. But what you can see here is that there's quite a large number of patients who actually did not have progression during this time period. And patients who had progression like Chrissy, who had progression at 9 months, so therefore she would have been one of these dots up here. Would have been a dot around here somewhere. She ultimately had further treatment and is still alive and in remission. And so therefore, this doesn't mean that these are the only survivors, but these are the only people who within the study didn't recur after their initial treatment as part of the study. One other caveat I should mention is that I'm going to talk quite a bit about prognosis. A lot of questions we received relate to prognosis, or in order to answer them, I have to discuss prognosis. And you'll see a lot of information about prognosis in these slides. So if hearing about prognosis is upsetting to you, please, be aware that we may be covering that. Now, one thing to keep in mind is prognosis applies to populations of patients. We're never really able to tell one patient what their outcome is going to be. There's always outliers, even in very good cancers and very bad cancers. There are outliers who have unexpectedly good or bad outcomes. So The other thing to keep in mind is these data apply to this population of patients and may not necessarily apply to you or your loved one. So please keep that in mind. We then looked, sorry, this is a longer time frame. So this, we cut it off at 3 years because that was the study time period, was the first 3 years. But obviously we were interested in what happens with patients long after that. And what's quite interesting here is at 3 years, the lines kind of flatten out. And what I mean by flatten out is we're not seeing recurrences. Because if you see recurrences are reflected by a little downward drop in the line, as you can see at these time points, these little dots are patients' last point of follow-up. So each of these dots means this is their last follow-up. So these dots here reflect people who are out 8 years. This person is out 10 years. So these dots reflect individuals. So, but as you can see, recurrences, really, we haven't had any recurrences after 4 years of those patients who've had more than 4 years of follow-up. And that's also a very encouraging finding that there really, there can be cures. There can be patients who make it to this time point who are truly cured. Now, the other thing that's quite interesting is the vast majority of our recurrences, more than 50%, happened in the first 12 months. And prior to seeing these data, I was of the belief, as many of us are, that early recurrences are bad. And I think that we've learned that's not true. And Chrissy is a perfect example of that. She had a recurrence at 9 months, and here she is 9 years later, cancer-free. So we previously believed, oh, if someone got a recurrence in the first year, their outcome was dismal. But that's clearly not the case. In fact, the recurrences between 12 months and 4 years only represent a relatively small proportion of recurrences. So people who are 12 months out without recurrences, their survival curve actually begins here. So their chance of being cancer-free a number of years later is obviously extremely good. And that's one of these concepts when we think about survival is, as you move along in time here, if you still remain cancer-free, your odds keep going up and up that you'll remain cancer-free. We also want to look if subsets of appendix cancer might behave differently. So we broke it up into the low, intermediate, and high grade, and you can see here a very big difference based on the grade. But again, these are not what we call statistically meaningful differences in the separation of lines here, and that's what this P refers to. That's what's the chance that this difference is real versus just by chance. And by chance, I mean if you toss a coin twice, there's a very good chance you'll get 2 heads. But that doesn't mean that if you toss it 100 times or 1,000 times, you'll get 100 or 1,000 heads. No, as the more numbers you get, the closer you'll get to the true difference. And in a coin, the true difference is either your heads or your tails. So this sort of helps us interpret the data with respect to the number of patients, the equivalent of the number of coin tosses. And this basically tells us that these are not statistically significant differences in outcome. And in each case, HIPEC, the line is the lower line. So if these were to be statistically significant, they would actually say EPIC is more effective or more effective than HIPEC. So there's nothing here signaling that HIPEC may be more effective. The other thing that, we knew that patients with low-grade appendix cancer did very well in the long run. I was surprised to see the low grades. All the patients that recurred in the first 18 months. No recurrences after 18 months, which is also something we were quite surprised, but I think reflects how much better our scans and our CEA and the other ways that we test patients for recurrences, how much better those have gotten, that we're identifying the recurrences more timely, because recurrence is actually a misnomer. There's no such thing as a cancer recurring. It was never gone. It was just not detectable. So these patients never really recur. They just go from undetectable to detectable. And the better our tests get, the earlier we can detect the recurrences. And I think that's what this is showing here, that even in the low-grade cancers that move slowly, we can detect recurrences relatively early. The other thing we allowed for is, I'm sure many of you have experienced a scan where there was a spot and we were unsure whether the spot was recurrence or whether it was just what we call post-operative changes, inflammation, scar tissue, or something else. And so what we allowed for in this study is if a spot showed up, let's say at the 9-month scan, if it never got any bigger, if it stayed that same size and never what we call progressed, then we didn't call that a recurrence. However, if at 9 months we were unsure, but let's say over time it got bigger and eventually got biopsied, or there was some proof that it was recurrence, we could backdate the recurrence the first time it showed up in a scan. So that really tells you the real time of recurrence, as opposed to sort of disregarding these indeterminate findings that allowed us to judge later what they were and then date back the recurrence at that time point. So, so it was true for all 3 types that recurrences tended to happen in the first 12 to 18 months. And what was also a bit surprising was this difference between the moderately differentiated and the poorly differentiated. Most studies up to this point lumped these together, and they separated low grade versus what's called high grade. And high grade really affects mod and poor. But as you can see, the outcomes are quite different between moderately differentiated and poorly differentiated. About 40-45% of patients with moderate differentiated cancer remained cancer-free in the long run, which was not true for the poorly differentiated. So very important, I think, that these groups get separated out when we consider the expected outcomes of treatment. So in conclusion, durable survival, long-term survival, can be achieved after cytoreduction for both well and moderately differentiated appendix cancer. However, based on the fact that the outcomes were essentially identical between the 2 arms, we believe that intraperitoneal chemo did not contribute to those outcomes, given the probability they're equally ineffective. Now, some people might argue, well, could they be equally effective? But if you look back to those, the results of clinical trials, and you look at the percent chance based on, if you look at the phase 2, back to the phase 2 outcome, where only 25% of phase 2 studies proved efficacy, proved one treatment was effective compared to a control. And in this case, we have 2 that were saying, if you try to make the argument that they're both effective, you got to multiply 25% by 25%, which leaves you with about a 6% chance that they work equally effectively, that both work, versus a 94% chance that neither one works. So essentially, in my opinion, the odds are very much that neither one was effective. And that certainly is supported by the data from the PRODIGE, the French trial in colon cancer, where they showed no difference. And it doesn't really dispute the findings of the original Dutch study, because that in retrospect, I look at that study and say the cytoreductive surgery is effective. It's just not the addition of HIPEC. I want to acknowledge a number of individuals who were key to this work. Andrea Sursek, who is my co-primary investigator, Floriana Marchetti, who collaborated with us at University of Miami. I have had tremendous, I started this as a junior attending, and I had tremendous mentors in surgery, including Pat Patey and Julio Garcia Aguilar and Ron Mateo, as well as in medical oncology, Len Saltz. I also want to acknowledge my the other leaders of our peritoneal program who were mentioned earlier, Yorgos Karagunis, Mike Foote, as well as my research team, Nikki Aguirre and Tina Gouda, who really helped do a lot of the data, the sort of the grunt work of the data. We also had fantastic clinical research coordinators. Any of you who are in EPIC probably met some of these individuals. These are largely young college graduates who are interested in a career in medicine. Alex was our first coordinator, and these were the ones that would call your oncologist to make sure you were getting your tests on time. They would also speak with you about the survey, the quality of life surveys we did. Alex went on to a medicine residency at University of New Mexico. Marty Morris became a plastic surgery resident at University of Chicago. Jackie Quinn is now a neurosurgery PA at Duke. Darius White is a med peds resident at the Harvard program. Jasleen Singh is now a clinical scientist for Merck. Mustafa Muhammad is currently in med school and will be graduating in a couple of years. And currently we have Chloe. And, you know, these people are just one of the privileges of doing clinical research at an academic center is you get to help develop the next generation of doctors, scientists, oncologists. And this is one of the very rewarding parts. There are many other collaborators and research staff who supported this. As I mentioned, this trial cost $2 million. So there's this TNTC means too numerous to count of other individuals. And I would be remiss without mentioning this trial clearly would not have been possible without the patients who agreed to be in this trial and who took that leap of faith and put their trust in me or their surgeon or oncologist to go through all this treatment. So I'm deeply grateful to all of those. And I know some of Chrissy, of course, and I recognize several other names on the audience here who also have been treated. And the philanthropists, all this, there was no government funding for this, all of this, and there was no drug company funding either. These drugs that we used have been around for decades, so no company was interested in funding this. So this was all sponsored by philanthropists who out of the good of their heart wanted to give their money to help get an answer to this question. And there were certainly large donors. The first donor who got us off the ground gave us $300,000, but there were countless small donors, and it all added up to make all this possible. So, you know, I can't express how appreciative I am to all of you. So thank you very much, and I'm happy to take any questions. And again, I've seen a number of these questions, and they do relate to prognosis. If prognosis is not something you want to hear about, you might want to disconnect. Hopefully, no one disconnects, but I certainly understand that people do. So we'll now go through the questions, and Chrissy's going to help with that part, I believe.
I am going to help with that part. So we're going to get to some of the pre-selected questions that we had talked about, but I do think there's one that came in while you were going over the results that I think we should just jump into since we're just coming right out of the results, which is, did any of the patients receive no HIPEC nor EPIC?
No, everyone in this study had HIPEC or EPIC. There was one patient in our, oh, actually, sorry, let me clarify that. There was actually one appendix patient. I was thinking they were a colon patient, but now that I think about it, there was one appendix patient who had a complication that thankfully resolved. And we felt comfortable giving her the treatment while she was admitted for that complication, while she still was admitted after the surgery and having recovered from the complication. But she felt that she didn't want to take any more chances. And she was randomized to the EPIC arm, I recall. Yes, because there was an option of giving it, right? So it could have been HIPEC. She was randomized to the EPIC arms. There was one appendix cancer patient who got neither. Now, what I can tell you is we are tracking our outcomes. Since the study closed, we've stopped using HIPEC or EPIC for appendix and for colon cancer. And we are collecting data on those patients. We were, you know, I was kind of wondering if the number of patients who might be interested being treated here might drop because we're not offering HIPEC or EPIC for colon or appendix. But it hasn't, interestingly. We still have the same volume of patients. So we're looking at those data. So far, there's no indication that outcomes have changed. Now, we closed for accrual 2 years ago, so there's plenty of patients who are still being followed, and there are plenty of patients actively followed on the trial and not on the trial. But that's something we're going to look at. But so far, there's no suggestion that the absence of HIPEC or EPIC has resulted in worse outcomes for our patients.
Okay, thank you for that. Can you do me a favor and drop your screen share so I can share my screen
Yes.
with some of these pre-submitted questions?
Yeah. And I can put them back up if people need me to show some of the graphs again. I'm happy to put the slides back up.
Okay, do we see Q&A here?
So emerging therapies, so new treatments. There's no new treatment that I'm convinced is effective. There are some that are, promising. I don't want to. HIPEC is there as the next section, so I'll address that is an emerging therapy, and I can talk with people about my thoughts about that. So putting aside HIPEC, I'm not aware of any new treatment with proven efficacy. There are, there's a number of things out there that look promising, but are still very much in the experimental realm. I am a big advocate for patients to consider clinical trials like HIPEC trials. I referred numerous patients to HIPEC trials. I don't think we can necessarily answer the efficacy question yet for that, because most of the patients that I'm aware of who participate in the HIPEC trials were at the sort of their last option. They were this was their last resort. They had no other options left. Their cancer was quite advanced. And, without treatment, their life expectancy was less than 6 months. So it's very hard. That's the right population to start with, because you have to start, you shouldn't be offering experimental treatments to patients who've got standard therapies. Anyone who has not tried standard therapies, and I'll explain a little bit about what I think is standard, shouldn't be considering trials. Trials are really for patients who are not eligible for standard therapies. Now, ICARuS was a little bit different because it was a trial of standard therapies. These therapies were broadly used and were used outside of clinical trials. So that's sort of a bit of an exception to that. So if, for example, you were offered a trial that used a standard therapy, but was asking a question like, for example, we've got studies looking at how if you can use fluorescence to identify peritoneal disease, or inject something to help the surgeon find the peritoneal disease during surgery. We don't know if it works, but it can be coupled with a standard therapy. So those sorts of trials, I think you should be open to. But I think if your doctor has told you there's no more options, trials like HIPEC, I think, are very reasonable. There's also a new therapy called Radspherin that looks quite promising. I'm not aware that there's a trial for appendix cancer, but I believe there is a trial for colon cancer. It's a therapy developed in Sweden, I believe, and it's using nanoparticles or microparticles to deliver tiny doses of radiation directly to the cancer cells. And the preliminary results look extremely promising. It's not published yet. We haven't really been able to dissect the results and independently interpret them. But the company is feeling very positive, and the investigators working, with the company are feeling very positive. And I and my collaborators have been working on something called PRIT, pre-targeted radioimmunotherapy, where we use antibodies to deliver tiny doses of radiation to the cells. So it's a similar concept, using something to deliver radiation directly to the cells, because you can't use standard radiation because, unfortunately, the cancer is so close to the bowel that you'd hit the bowel with a lot of radiation, and bowel doesn't tolerate radiation. So the dose that you would give would be limited, not enough to kill the cancer because it'd be too toxic otherwise. But when you deliver it, where both I and the company that developed Radspherin, they're looking at administering it in such tiny doses and localizing it right to the cancer cell that you can hit the cancer cell and a small radius around it. So we think chemo doesn't work as much as we would like for cancers, including appendix cancer, because some cells are resistant to chemo and it kills only a proportion of them. So those people who thought they were cancer-free and their cancer came back, it's because there was enough microscopic cancer cells that survived. Now, the concept with both the Radspherin and the PRIT is that you're creating a radius. You're like detonating an old nuclear bomb, and you're killing cells around it, but very, very close to the cancer, which is mostly other cancer cells. So we think this might be really, this approach might be the way forward, as opposed to what we've been doing, which is largely putting poison into people. And that's what chemotherapy is they're poisons. The first chemo was developed from mustard gas, which was used during World War I to poison people. These are really poisons that you can manage to give in doses small enough that they don't kill the person, but they kill the cancer. But this is a whole new level of how to deliver therapy with minimizing toxicity. So ours, they've done in humans with promising results. Ours have been just in animal studies so far, but with also very promising results. We're curing appendix cancer in mice right now, which I realize is not, is a long way from curing in humans. But we've also shown that it's safe, or at least our preliminary data show it's safe in primates, in monkeys, which is sort of the best model of toxicity in the animal world. So we think these are promising things coming down the line, but not yet ready for prime time. There may be other drugs out there that you may hear about. They may, if it's in setting of a clinical trial, I definitely encourage it. I would not encourage people to consider these experimental treatments if they're not actually part of the trial. To me, that's, there's a lot of danger there. And generally speaking, with few exceptions, there's, questionable ethics of it too. So efficacy of HIPEC, let me get back to what's the best treatment now. It depends on the type of appendix cancer. Generally speaking, low-grade appendix cancer, surgery, and I'm only talking about stage 4, but surgery, cytoreduction, and at this point we do cytoreduction alone for low grade. And then for intermediate or high grade, typically we give the systemic chemo, which is Folfox, or in some patients FOLFIRI, or in some patients FOLFIRINOX, as the first treatment, and then surgery, and then typically surveillance. Most clinical trials that involved chemotherapy after surgery, so systemic chemo after surgery, we're only able to actually administer the systemic chemo in about 80% of the patients. About 20% just weren't up for chemo after their surgery. So by doing chemo before surgery, you have the advantage of ensuring that you get it. You tolerate it better, generally speaking, than when you're recovering from surgery. And also, we get a sense of whether it's working. For low-grade appendix cancer, the efficacy or the amount of the number of patients who their cancer will shrink on chemotherapy is about 30%. Whereas for intermediate grade, for the moderate differentiated, it's about 70%. And then for the poorly differentiated, which includes Signet and goblet cell, it's about 60%. But if you give the chemo after the surgery, you've got no idea if it's working. Our best surrogate for working is that the cancers will shrink. So generally we favor, if you plan systemic chemo, doing it before surgery. And that's sort of what our standard approach. There is on the MSK website, you'll actually see Chrissy's story, and also there's a lot of information about our approach to appendix cancer. So if there's things I didn't answer today, I definitely encourage you to go to our website and review. There's several pages that talk more about this. So my opinion on efficacy of HIPEC is I think it's unknown at this point. Certainly for patients who have other options, I wouldn't encourage it. But if you're out of options, I would definitely encourage you to consider being in a clinical trial. We're not performing it here. My suspicion is that if there's benefit, it'll be only incremental, small benefit over what we're doing now. And we're putting most of our energy into developing new treatments that might really have a much bigger impact. And, because we have limited resources, we don't have unlimited amounts of money. We are putting our efforts in developing new treatments, as well as trying to understand what's causing appendix cancer. We're very interested in lamins. We've started a lamin clinic now. We generally see over 100 lamin patients a year. And these are patients who don't have evidence of spread of the cancer. And we're trying to look at them as hopefully to give us a sense of why some people develop cancer and some don't. So that's really where we're putting most of our energy and effort into. But again, it's not that, I wouldn't discourage any patient from pursuing it. But again, we're just pursuing other things. Goblet cell. So there were about a quarter of the patients in the study, approximately, were goblet cell adenocarcinomas. So the results look no different for them than for all the other patients. So we're not using HIPEC for goblet cell. We're using typically Folfox. And if there are candidates for cytoreduction, we discuss cytoreduction. So this is a very tough question. I have goblet cell, and my surgical oncologist questioning whether it moves forward with cytoreduction, HIPEC, or low-dose chemo. What are my options, and how many years will I survive? So it's very hard with just that, brief amount of information to answer that question. In my mind, if you're a good candidate in general for surgery, you're a healthy person and you get through potentially a big surgery, if the burden of cancer in you isn't overwhelming, if it can be done with an operation that won't just destroy your quality of life or be extremely high risk, I think cytoreduction is reasonable. But I would do it coupled with systemic chemotherapy. Surgery, patients I've seen, excluding the well-differentiated. So if you've got moderate differentiated or high-grade goblet cell, and chemotherapy doesn't shrink your cancer, the chance surgery will make a difference is close to zero. It's probably lower than the risk of major complications. In general, I found that if chemo doesn't work, surgery is also not going to work. So I typically, for patients who've not had chemo, I'd recommend Folfox, FOLFIRI or FOLFIRINOX, one of those combinations, and there's different reasons why patients should take them. So I couldn't tell you for you which one, but I generally favor systemic chemotherapy. And if you have a response and you meet all those other criteria I mentioned, cytoreduction is a reasonable option, but I would not recommend HIPEC because my interpretation data is that it's not effective.
Okay. So those were some of our pre-submitted questions that we sort of grouped together because we saw some theming there. We do have several questions in the chat, so I'm just going to kind of throw them at you. The first one being, if neither EPIC nor HIPEC is effective, how do patients find the best CRS surgeons for appendiceal cancer?
So I would encourage you to ask about their volume. How many of these cases do they do a year? And how many years have they been doing it for? There's very good data showing that complication rates go down. One of the most compelling ones is that Dutch trial I mentioned where 8 patients died from complications of surgery, and those were their initial experiences. But as they got more experience, their mortality dropped dramatically. So you want to know how many they've done and what their mortality rate is and what their complication rate. In general, complication rates are high. They're 30 to 50%. But it's more about saving the patient from the complication. So we've had out of the last 1,000 cytoreductions we've done, we had 1 patient death from complications of the treatment. So we have a 0.1% mortality, and that 1 patient was in ICARuS. So you want to make sure that your surgeon has a low complication rate and a low mortality rate and a lot of experience, because experience not only lowers complications, but most likely gets better results. You're more likely to get a CC0, and they're also more likely to use good judgment because so much in this field comes down to the surgeon's individual judgment of what to do in the surgery or leading up to the surgery. So the more experience they have, the more they've got to base their judgment on. So they're more likely to make a good decision for you about, for example, you go in for cytoreduction, and sometimes we have to make a decision whether we should proceed or not proceed with the surgery. And that really is a judgment call influenced by a lot of experience. So experience. The other thing is where they work and what is the reputation of that hospital. There are a lot of people out there offering the surgery in community hospitals and a lot in academic centers. There are some community hospitals that offer, excellent interventional radiology, excellent ICU care, the other specialists that help support the care of the patient. So you have to also look at the larger context. Also, a lot of studies have shown that academic centers where a fellow or resident is involved in your care produces better results. It was a very compelling study. If you had a heart attack and you were admitted to one of the Harvard hospitals, it was a study from one of the Harvard hospitals, and you got admitted to the resident-run service versus the private attending service, you had a better survival chance if you got admitted to the resident service. And many people are like, hang on a second, why would I get a better result with a resident than an attending? Well, those residents are supervised by attending. So you're actually getting a team, including attending with all their judgment, who'll be there doing the procedure. But you've got not just a nurse following you into the hospital, and not just the surgeon coming to see you once a day. You've got 24-hour coverage by residents. So there's a lot of reasons to believe that academic centers can offer safer care and greater salvage or saving patients from complications. Because my complication rate from this was around 40-something%, but the mortality rate was 0.1%. So it's about saving the patient from the complications, and that's where having the residents and fellows support that is, critical. Thankfully for me, I work with fellows, and for my patients, and my fellows have typically gone through 7 or 8 years of training, and they're kind of finishing their training, so they're quite experienced in helping manage these patients. So these are the sorts of questions that you should be asking. And I
To be
think
clear, nobody was good at managing me except for you. So even though you kept bringing all those people in.
Well, they're helping in the background because they're the ones often who deliver the message to the attending and help, say, hey, listen, this patient, their labs show this, and the attendings, if you're in the surgery all day, you're not seeing that abnormal kidney function at 10 o'clock in the morning. You may not hear about it till much later in the day. Whereas if you've got a team who can jump on these things, like let's say you have a fever and you need a scan and there's an abscess, you want to make sure there's a team. I'm not managing the abscess. We've got infectious disease doctors recommending antibiotics. We have interventional radiology doctors doing procedures to help manage these things. So yes, you want that the surgeon to show their face and be the face of the team, but you also need that team. So.
Hey guys, I'm sorry to interrupt, but I just want to do a time check. We got about 17 minutes.
Yep.
And I have been seeing lots of questions come in over the Q&A area, and I've been adding them in, Chrissy. And
I have them.
oh, good. I just wanted to
Yep.
make sure. Okay, so if y'all want to use this time to kind of churn through some of these, and maybe if we can't answer them all here, maybe Dr. Nash would be willing to answer them, um, via Google Sheets or something.
I'm sure we can come up with ways of answering questions outside of this forum, and some can be answered simply with just typing an answer. Others are more complex, but I'm certainly open to doing this. And I've also talked with Amanda and Chrissy about scheduling future events to talk about appendix cancer. The theme might change, but there'll be other opportunities to ask questions as well. So
anyway, I'm gonna say this one last time. Thank you, thank you, thank you so much.
My pleasure.
So in the Q&A here, why do you believe other renowned appendix cancer centers are so supportive of HIPEC, and do you expect this to change based on your results?
Well, a lot of centers have changed. So with colon cancer, because those data have been out for a while, ICARuS, the appendix cancer data is not published yet. I presented it at meetings and I've been talking about it, but I haven't published the paper yet. We're working on it. You want to make sure you've got every detail correct. So we're still working on that part. So part of it is because it's relatively new information, but there's a natural bias, just like there's patients here who probably, have been treated with HIPEC or EPIC and have had good results and believe that helped them. Lots of surgeons believe that too. Surgeons want to believe they're helping people. It's very hard to accept the fact what you may have done to a patient didn't help them. And there's lots of this retrospective data. So hormone replacement therapy for postmenopausal women was the #1 prescribed drug in the United States for a long time. And it was based on these retrospective studies, large studies, often with tens of thousands of patients, that women who took hormone replacement had fewer heart attacks than women who didn't. And so the conclusion was that there was some protective, that the hormones were protecting women against heart disease. Finally, the randomized trial was done. And not only did it not actually protect women from heart disease, there was no benefit in that regard, but more women died of ovarian and breast cancer who were in the hormone replacement therapy arm compared to the placebo arm. So it was actually a harmful therapy, and now the use of that has dropped off massively. But that's a classic example, and there's many others, of things that were thoroughly ingrained into the medical literature and into clinical practice that turned out simply to be not true. But it's hard to accept those things, and you can always pick apart trials. The French trial, the Prodige study, used oxaliplatin. So people said, well, maybe mitomycin works, so let's keep working with mitomycin for HIPEC. Our trial now undermines that. But it's hard to change beliefs, and it takes time. But there are a number of centers. I was at a panel with someone from MD Anderson, someone from Brigham and Women's, and I think Cleveland Clinic, or no, Mayo Clinic. And we were all asked, it was about colon cancer and peritoneal disease, but we were all asked about what our standard is now for colon cancer, and none of us were considered IP chemotherapy a standard therapy for colon cancer. We've stopped using it. But these things, practice changes slowly. I can't read into everyone's psychology, but a lot of this is psychology rather than science.
Another question, what do you recommend for Lamin patients with recurrence? And also, is MSK going to consider offering EPIC or HIPEC in the future?
We have no plans currently to consider either. So Lamin's, our experience has been that patients who are initially diagnosed with Lamin, sometimes with mucin in the abdomen, but no cancer cells in the mucin, because that would be cancer, not a lamin anymore. We find that 95% of those don't recur. We're now offering surveillance for all those patients, and we've found that those numbers are holding up. And the 5% who recur, we've managed to successfully treat and render them NED again. So generally speaking, we offer surgery to those patients. As long as there's no indication that it's progressed into a higher grade, we don't typically offer any chemotherapy, just surgery alone.
Okay. Have you seen a significant difference in outcomes between those with signet originating in the colon versus those with signet originating in the appendix?
Good question. They're both, unfortunately, aggressive types. And there's two types of signet in the appendix. There's signet that arises from intestinal type and signet that arises from goblet cell. So there's actually three different types. We treat them similarly with respect to the type of chemotherapy and the surgical approach. And so far, the outcomes seem similar, but you need to do a pretty large study to determine if they were truly identical or whether there's some difference.
Okay. Is any immunotherapy effective in treating appendix cancer?
So far, no, although some of you may have heard that we expect to open an immunotherapy trial before the end of the year. We've got, there's still some logistics to work out, but we have funding and we have the drug. It's BAT-BAL, the combination therapy. None of the prior immunotherapies have been effective for appendix cancer, largely because when in GI cancers, immunotherapy largely works for the mismatch repair deficient ones, the MMR deficient or the MSI ones, and we don't really see MSI in appendix. We do see it occasionally in colon, but very few actually metastasize to the peritoneum. I've had, I think, a couple patients who got immunotherapy for metastatic colon to the peritoneum. They had mismatch repair deficient type, the type where it can work, and they actually, when we took them to surgery, all the cancer was dead and they appeared to be cured. So it can be very effective for mismatch repair deficient colon cancer, but as of now, it's unproven for appendix and for non-mismatch repair deficient colon. So like I said, we are opening a trial later this year, and you'll see it on our website when it comes open. And that would be for any appendix, pretty much any appendix cancer who's gone through standard therapies would be eligible.
Okay. Something that's talked about a lot in the groups that I see, which is what type of test patients should get to check for recurrence for any type of appendix cancer.
Yeah. So we generally rely on CT and CEA, the tumor markers, sometimes CA125 or CA199. We check them at baseline if we can before we treat the cancer, so we know if it's elevated, because if it's not elevated initially, it's not likely to be helpful later. These are proteins that are detected in the blood. So we generally, CT is our standard, and then CT scans. MRIs are appropriate if there's appropriate expertise. MRIs can be a little more tricky to interpret, so you need people who really got expertise in MRIs for peritoneal disease if you're going to have MRI. But generally, my colleagues here believe that CT or MRI are equally good. I prefer CT just for my own personal reasons, because I'm much better at interpreting the CT, and often I see things that sometimes even the radiologist occasionally misses on the CT, or at least I can discuss it more intelligently with them. Whereas MRI, I just don't have as much experience for rectal, or other than in rectal cancer, which I also treat with MRI. I'm quite good with that, but not in the peritoneum. So ctDNA is an interesting area that we're exploring. We're not quite sure how useful it will be, but we've certainly seen some patients who it was a good predictor of their recurrence, and it was, they had to go up, they went on chemo, it went down, they had surgery, it went to nothing, and then if it recurred, the ctDNA went up. So ctDNA is, I think, ultimately will be helpful, but we're still learning how to use that.
And there's been a lot of talk in some of the support groups that I'm in about the articles that circulated earlier this year about radiation from CT. So what are you telling your patients about radiation concerns?
So certainly with children, it's a major concern, radiation. Appendix cancer is not common in children, but can occur. So we very much try to avoid CT in children. In adults, the risk is small. It's not zero, but it's small, and it's over decades. So, if you're facing a cancer that has a significant chance of killing you, the chance that you're going to get a different cancer from the radiation from a CT scan and not killing you is very, very small. So if CT is sort of the thing that otherwise makes most sense for you, I wouldn't be concerned about having a CT scan. But again, if you're any patient who tells me I really don't want a CT scan, do I have to have it? We do MRIs, or we alternate and do CT versus MRI. But honestly, the risk is it's there, but it is quite small. Again, it's helpful for populations, right? So for populations, we see a certain rate of cancer. When you look at individuals, it's much less of a factor.
Okay. Question, do you think HIPEC is harmful or just ineffective?
Well, the PRODIGE study, which is the only one that distinguished, where you had HIPEC versus a placebo, essentially, showed there was a higher complication rate with HIPEC. In my mind, the biggest concern with intraperitoneal chemo is it can magnify a surgical complication. So while certain complications like infections may not be caused by the IP chemo, it certainly could make you a more complicated infection. So my feeling is that the data is quite clear that it increases complication rate. So it's not just a freebie, and it adds about 3 hours to your surgery, which is not trivial because these are long surgeries, and the longer someone's in anesthesia, particularly if it goes above 12 hours, then the hours really can quite significantly increase your complication rate.
Can you explain why a PET scan isn't normally recommended for this disease?
Well, most appendiceal cancers are mucinous, and mucin is a byproduct. It's actually an inert. It's not cancer. It's a product of the cancer. It certainly causes problems when it builds up in large amounts, but because it can compress the bowel. But it's not cancer itself. And because PETs light up when a cell is using a lot of glucose. So your brain uses a ton of glucose. The brain always lights up massively. But slow-growing cancers don't tend to light up that much. and cancers that are cancer cells floating in a bunch of mucin aren't going to show up. And the resolution, the ability to find small things, is no better for PETs than it is for CTs or MRIs. In fact, it can be worse. The time I find PET helpful is if occasionally you see a spot, a nodule, and you're trying to distinguish if it might be cancer or not. Sometimes PET can help, but probably I would say, PET accounts for only a few % of all the scans I order. There are other cancers where it's helpful, but not for appendix, or even in fact colorectal, because that's also pretty mucinous in the peritoneum.
Okay. This person, the way they phrase their question is, how much CRS can one person take when this is the only option? So I think that's like, how many surgeries can one person have if that's the only treatment option?
Yeah, I mean, not all, CRSs are the same, obviously. If someone has a CRS where they've had their spleen, their colon, their rectum, their gallbladder, their uterus, their ovaries, if all of those things are removed, that's a very different situation than someone who's had an appendectomy and had some stripping of peritoneum. It's a very, I'm a firm believer that all care has to be individualized in this realm. We need to, yes, we have guiding principles, but you have to look at each individual case. So I never, occasionally I say to myself, I never want to have to do another surgery in this person again. This was so high risk and difficult. I don't think they could ever go through another surgery. And then they find themselves in a situation where there might be some benefit of surgery, and I reconsider. So I try not to ever close the door to consideration for cytoreduction. I think some of the sort of absolutes in my mind are, if the patient is bedridden, for example, if they're so weakened from their cancer or their complications or whatever, that's not someone who should go through cytoreduction. Also, if a lot of bowel is involved, that's also a problem, because sadly, most patients who die from metastatic appendiceal cancer die from malnutrition. Their bowel fails, or they get obstructions and they get malnourished. And we don't really have a good substitute. Using intravenous nutrition has not been really shown to prolong lives of patients in that situation. So if there's extensive bowel involvement, that's the part that's going to kill the patient. So if they're going to die of the cancer, so you don't want to take out a bunch of bowel and already contribute to malnutrition from the start, so to speak. So I'm very careful around that. And then, and otherwise, what's the patient's willingness, So I always try to keep an open mind. So there's no number for that.
We're almost at time. I'm going to give you this last one, which is, have you heard of any success with ivermectin or any other off-label treatments? I see this all over the internet.
I mean, ivermectin has sort of had this life of its own with zero data. There's some small mice studies, but it is so far from anything that a person with a scientific mind would consider reasonable. So there's all this stuff out there, and ivermectin can be harmful. So if someone tells you blueberries will help, I have no problem with you eating blueberries, because blueberries are good for you, right? So but chemicals, I would be very, very careful. And I almost never, I don't think I've ever recommended sort of an off-label, unproven chemical like ivermectin for a patient. If you've got worms and you're a horse, it's a great drug for you. But if you've got cancer, I couldn't recommend it. So.
Okay. Well, we are like less than 1 minute away from 8 o'clock. So I'm going to thank you so much for your time and for sharing these results with us. We have some more questions that we didn't get to. So for the group that's still here, we will work with Dr. Nash to get some answers to those. And I know Amanda is going to make this video available, and we will share that in all the groups where we've been sharing registration. But thank you so much for